I'm so tired. Oh. And I just feel my phone.
And you guys should have all your grades for now, sorry. Any questions about before we start today with class? Any questions on any of the exam questions? There were a couple of them that I saw getting missed. A lot of you, some of you were on the right track, so I gave partial points for that question with the, the protein. I gave you the structure of the protein and then asked you what happened if the signal sequence was not there or what happens if nothing happens. So the one that we had given you, we talked about, so if the signal sequence is not there on that protein, so it was a membrane protein, right? It eventually had to go into the membrane outside the plasma membrane. So if the signal sequence is not there, what would happen to that protein? Where would it go? So that's one possibility, that it would stay in the cytosol. I also gave points if you guys talked about how it may have the start sequence for the ER, like localization, and it would go into the lumen, but it won't get out, right? So I gave partial points for that as well. But the correct answer would have been that it would have never made it anywhere because it lost the signal sequence. It would have stayed in the cytosol. What was the other choice in there? What were the other questions? Another one in there was, so it was an N-linked protein, right? It was tagged at the amino end with the GFP. All the different mutants that we had, they were all gonna be tagged with the GFP. So just because, so there was this one in there that the first 15 amino acids are cleaved, right? In one of the mutants, it talked about that on the N terminal, the first 15 amino acids are not there anymore. Well, the GFP's tag would still be there because you put it on. So if you wrote that no GFP would be seen because the tag won't be there, well, the signal sequence will be gone, but the GFP tag would still be there. And the reason you know that the signal sequence will be gone is, remember that chart that I showed you when we talked about intracellular transport? And there was this chart that talked about the different sequences, the nuclear localization signal, ER membrane, start signal, stop signal. In there, for the ER localization signal, where it actually comes and gets into the translocator on the ER membrane, that's in the first 15 amino acids. So it has like methionine, a couple of amino acids, and then the signal sequence starts. So that would have been lost. So again, it would have been in the cytosol in that case as well, because it wouldn't have made it in. If the transmembrane domain was deleted, where would it go? Yes, it would still make it into the ER, but it won't be able to then go into the membrane because the transmembrane domain, which allows it to be in interaction with hydrophobic environment, would be lost. So it would go into the lumen. Good. What else was there? There were four. Oh yeah, the ligand binding domain. So if the ligand binding domain was messed up, was mutated or deleted, what happens then? Does it affect the GFP localization or the protein actually going where it needs to go? Yes or no? I had some students that were on the right track with it. Some kept talking about how the ligand binding. So ligand binding is activity of the protein eventually, right? So that's most likely, for example, if it's a plasma membrane protein, it's most likely acting, for example, as a receptor, right? That's one possibility. So the ligand binding would be where it's binding to that molecule outside as a receptor. So that shouldn't interfere with its localization signal. Localization signal is completely separate from where the protein's active site is. So it would still go into the membrane and it would still act it would be the normal GFP signal that you would expect to see, but its activity would be affected. It won't be able to work the way it normally does. Okay? Yeah. That's clear. Any other questions on there? This one also I don't. You guys are being quiet. Okay, so we'll go ahead and get started on gene expression control. Last week, we started talking about different ways that gene expression can be regulated, right? And we talked about 7 different levels at which it can be regulated. And we've been talking right now, most of the time last week, we had been focusing on how it was regulated at the level of just the actual gene transcript, right? When it gets switched on or off. So it was talking about that promoter region a lot. We talked about the operator region and the promoter region. We talked about activators and repressors, right? Inducers and co-repressors. And we looked at 2 different examples. One was of the lac operon, and then which was the other one? We looked at one other one. So we had the lac operon and the tryptophan biosynthesis. So in the tryptophan biosynthesis, we talked about how it was controlled by what? Most of that was getting done at the transcriptional level, right? Looking at just the gene transcript, we had in the operator region, we had that repressor site where it was binding on there. And when there's no tryptophan present, it will not be able to bind and you will get transcript to be made. That was one level of control in there. What was the second level of control beyond that? So one was just simply, hey, is tryptophan there or not? If the tryptophan is there, it binds to the repressor. It allows it to bind to the gene promoter region, right, the operator region, and it shuts it down so that it's not transcribed. And then when there's no tryptophan there, the repressor just floats around. It cannot bind to its operator region and it cannot shut down the transcription, so the transcript will be built. There was a second level of control in the tryptophan. What was it? We had those fun little structures that we talked about. What were they? Stem loop structures, and that was at the level of what? Not on the gene level, but where? Say it again. So it was with the, at the transcription or like a little bit past transcription was the mRNA level, right? And it was the mRNA transcription to translation portion that affected it. So in there, if you remember, this is tryptophan operon has like all these different genes that are part of it, right? A, B, C, D, E, that are all on that operon, controlled by the same, promoter region, right? This is then the operator region where the repressor comes through. Well, this little transcript, right? So actually, let me squeeze it up a little bit so I can explain this better. We talked about how, so this would be the start side of transcription. And then down here somewhere, well, on here it would be A's. So it would be transcribed. Here somewhere would be your end site, right? So over here, you have the start site and the end site. These little regions were the 5' and 3' untranslated region. Remember we talked about those? The 5' UTR, 3' UTR, right? Those are the untranslated region. And these are not without reason. Sometimes they also act as regulation, right? They were another level of regulation. So what was in here that controlled whether tryptophan was going to be able to, this operon was going to be translated or not? What was present in this 5' untranslated region? Huh? Yes, so there was this, if we would call it URF because it was in the untranslated region, it is an open reading frame that gives rise. So in here somewhere is a little transcript with its own start and end site for a leader peptide. That's what it was called. And this leader peptide basically was another level of control. It was able to do this, just create the leader peptide because it was in a different frame than the rest of the operon, right? And so in here, you got the leader peptide and some other mRNA sequence as well that was going to be produced. And they created certain stem loop structures that would control whether or not you got the rest of the operon made, right? And I'll actually go back to it just for a second, because I want you to appreciate that. It's very important for you to understand. So basically, all this structure, this is in that untranslated region, okay? None of this is in the actual operon region. All this structure, this is all in the untranslated region. And what you get are, at the beginning of this transcript, in that untranslated region, you have a start codon, and then soon afterwards, you have two tryptophans that are coded into the transcript. And those tryptophans are going to be what controls whether or not this is going to be made. If there is lots of tryptophan around, right, tryptophan is present, you're going to end tRNA with tryptophan present there. So they'll be able to go through this very quickly, right? It can put in the tryptophans where needed and continue to translate down the road. When that happens, there is no chance for the 1 and 2 region to bond together, right? And there is going to be no chance for them to create that first stem loop structure. Instead, only the 3, 4 will be able to bond and create that structure. And that will signal that, oh, there's enough tryptophan around, we don't need more. Clearly, because we were able to fill that in. And this will allow the actual transcript to be made. On the other hand, if there is no tryptophan around or not enough is available, the ribosome is going to be stuck on that codons, right? Waiting for tryptophan to show up, the tRNA for tryptophan to show up with tryptophan so that it can continue. So because it is stalled, it's going to allow for region 2 and 3 to stem loop together. And this is going to then kind of stall this ribosome over here, and the rest of the transcript is going to be able to be made. Okay, so it's just controlling whether or not a ribosome can jump on the actual operon and transcribe it. Yes? No? So that's something that you want to be mindful of. And then the second one that we talked about was the lac operon. And in the lac operon, now there were multiple regions of control. You had a repressor, you had an operator region with a repressor, and you had an activator, right? So looking at the actual operon itself, you have the main operon starting down here. You have your -10 and negative 35 side with the Tata box and everything. You have the operator region. In that operator region, you have the lac repressor that binds in this first one. And then upstream from the -35, so further upstream from there, you have another promoter region that has, or another operator region that binds the activator for lac operon. And it's a balance between whether there is glucose present or lactose present or both present that are going to control whether lac operon is switched on or not. So the purpose of the lac operon is what? What's its function? What does the lac operon do? Yeah, so it's basically lactose, right? The tryptophan operon's function was to create more tryptophan when needed, right? As an amino acid. This one, the function is to be able to process, metabolize lactose. Well, if you have a lot of glucose around, you don't need to metabolize lactose, right? Because glucose is the more efficient energy source for you to utilize. That's what you will always go to first. And in that case, you will have a lac repressor that is going to be bound to your operator region, keeping it shut off. And that is going to be present whether or not there is lactose there, right? It's going to just keep it shut off because there's nothing there. Now, when you have no glucose and no lactose, you're going to have the lac repressor bound to the operator because there's no lactose, but you will also have there's cyclic AMP bound to the CAP activator and bound to that second region as well. Now, this by itself would have activated lactose metabolism, but because there's no lactose, the repressor is gonna be bound there too, and that's going to keep it shut off. The only time you're going to see activation would be when there's enough lactose present to shut down the lac repressor, so lac repressor is not bound, and there's no glucose present. So there is enough cyclic AMP around, right? Because there's no glucose there to create the ATP to bind to it, so you have high levels of cyclic AMP, and they are bound to it, the activator, which will then recruit the RNA polymerase and actually move it in the right direction and create the operon transcript so that you can get lactose metabolism. Yeah? Cool. Yes.
So you said for the CAP activator. And APN can do lactose or no?
So yeah, so the CAP activator is going to activate the replication, the transcription of this operon. But it can do it alone, right? To be able to bind to that activator region, it has to have cyclic AMP bound to it. That's gonna be the one that activates. It's the activator essentially. So this construct, even though this is CAP activator is always around, it cannot bind until cyclic AMP is there to bind to it, and that activates it and binds it to the DNA sequence. So that's part one of it. The second is it alone cannot recruit RNA polymerase if there is lac repressor bound further up, because polymerase won't be able to settle down with this blocking the way. So you have to have the lac repressor off the genome before the polymerase can bind. Okay? Yes.
Actually, so the lactose that's what allows lactose to be synthesized for.
So lac repressor is not gonna allow for things to be, for the operon to be synthesized.
Yeah, there's no lactose there, so it's only there. So it's just, is it what causes the lack of lactose?
No, so when there is no lactose, there's no lactose to synthesize, yeah. And so the lac repressor is gonna be hanging on to the genome at that time and during that process. Typically, again, the lac repressor is always present unless it is recruited off because there's a lot of lactose around. The presence of lactose causes inhibition of lac repressor transcript to be built. So that's a control mechanism for the actual repressor to be made or not. Does that make sense?
I guess, yeah, it's kind of, if it's only there when it's a lack of lactose, and it's repressing, I'm just kind of confused with its actual.
No, so it's gonna be there, whether, when there's no lactose, it's gonna be transcribed and it's going to be present. And when it's present, it's gonna be bound to the genome.
Yeah.
If there is lactose present, it's going to inhibit production of lac repressor. So it's, the transcript won't be made, it's not gonna be translated, there'll be no lac repressor to bind. And so the only thing that will be present then would be the activator. And because there is lactose around, you can now actually have something to metabolize by this. So you're not gonna make this transcript unless you need it. Does that make sense?
Yeah.
So lac synthesizes lactates.
Mhm.
Enzyme, okay. Sorry.
Okay, so today we're gonna go to the next step of regulation. So we are going to now look at the genome level, not just at the gene level, but at the genome level, how we are going to regulate the transcription happening for particular genes. And we're gonna talk about chromosome modifications. So some of these we've actually already kind of talked about when we talk about DNA structure and function. And we are gonna continue on that. And we will touch base on them more. I got a new one, where is it? So I don't have to walk back and forth. And be lazy in front of the screen. Watch me lose this in 2 weeks too. I keep buying new ones and then losing them. Now, it's only my second one this semester. Yes. I've been better this. Okay, so let's talk about that. So when we talked about DNA structure, we talked about how DNA is packaged inside the nucleus, right? And we talked about how you have those histone octamers and the DNA is wrapped around them, right? And those histone proteins, they have these little tail regions that at the amino terminal that can be modified as well. And those are typically modified by additions of what small molecule? There were two different types of modifications we talked about, and we'll see them in a second as well. Methylation is one of them, and I heard the other one too.
Phosphorylation?
No. Yes, they can be phosphorylated, but that's for a different control reason. But yes, acetylation. Oh, sorry. There you go.
Period. You don't get us there.
She's like, I'm I said it, not her. Anyway. So yeah, so you have two different types of chromatin when you look at how well they're packaged or how tightly they're wound up and packaged inside the nucleus. One of them is euchromatin and the other is heterochromatin. Euchromatin are all these kind of open regions that allow the DNA to be accessed by the transcriptors, by other proteins to do the work they need to do, including transcription. And heterochromatins are regions that are very, very tightly packaged so that you cannot access them very easily. And they are the ones that are going to be what we call silenced region of your genome. These regions, there are some that are always going to be in this heterochromatin form, right? So centromeres on the chromosomes, those are going to always be in a heterochromatin form. Your telomeric regions at the ends of the chromosome, they are always going to be in a heterochromatin form. But then there are other regions that can change based on developmental stage or the type of cell you are or the environmental control. And sometimes they will be in heterochromatic form and other times they will be more open in euchromatin conformation. So in this heterochromatin, basically what you are talking about are this constitutive heterochromatin and facultative heterochromatin. So the constitutive are these regions that are always bound tightly, and facultative will be based on what you need, right? Sometimes it will be closed and sometimes they will be open. The way that happens is through modification on the histone tails at specific, lysine and arginine residues usually. And these are in the form of methylation or acetylation, occasionally phosphorylation. Now, typically, methylation means leads to more tightly packed chromatin, and acetylation leads to more open conformation. But it's not a hard and fast rule that acetylation will always end up being open or methylation always closes up. The reason is that usually you'll get multiple types of modifications happening on any given region. And it's the overall change that happens when you have all of those combined together that will dictate whether that area is closed or open. If it was just methylation alone, typically that leads to gene silencing and that area is going to be closed up. If you have acetylation and methylation, it can lead to somewhat open conformation and some level of gene transcription happening in that region. Other times you have regions that are not methylated, but acetylated and phosphorylated. These are usually regions that are really, really open and really active in your genome. So phosphorylation definitely kind of opens it up. Acetylation also tends to be more open conformation. So when both of them are present, typically that's a very loose chromatin and has lots of things that are happening in that region. Okay, so where is that important? Now, this is important in everyday, just like general developmental process as well. And these are some very important applications of it outside of just gene regulation at a single gene level. So if you look at X and Y, your sex chromosomes, if you are a female, you have two X chromosomes, right? And if you're a male, you have an X and a Y chromosome. Well, if you have the X and a Y chromosome and you compare the gene expression of all the genes on the X chromosome to that of a XX genotype, you're going to have half as much expression, right? Because you only have one copy of that instead of two. Well, that would not be a big deal if there were only sex-related genes on there. But that's not the case. Our X chromosome actually contains lots and lots of genes that are important for everyday function as well. And you cannot have twice as much of those transcripts in one case versus the other. So there has to be a way to control and regulate and kind of normalize that level of expression across the board, regardless of whether you have XX or XY genotype. And they do that by in early embryogenesis, just when the first zygote is formed, and as it starts to starts to develop and starts to replicate and divide, you have inactivation of one of the X chromosome in each of your cells, and this happens randomly of sorts. So you can have it where only one particular X chromosome is selected across the board, and that's the one that is shut off. Or you can have inactivation of just random X chromosome. So initially in that early embryo, you will have this happen at one point where half of them, if you just look at just probability, about half of them will only show the X phenotype from the mother, and half of them will be the paternal. The other X chromosome is heavily methylated and shut off. And so this one is completely inactive. We have found that there are, even in that inactive state, there are few genes that are still active within that as well. But in general, this is going to be heavily methylated and closed off and just kept that way. And then after that first level of random selection happens, every cell that derives from those original cells are all going to inherit the same X chromosome inactivation pattern. And so it's always going to be the same that's going to be inactivated in every subsequent replication, okay? Now this can be seen actually inside the female nuclei as this little dark, very heavily stained spot. And we call this bar body. So you can tell where that is present inside that nucleus. Now this means that any XX genotype individual that you look at, regardless of which one you're looking at, what type of animal or what type of creature you see, is going to be a mosaic, right? Because half of their cells usually, will have one type of genotype and the other will have the other, since this is completely random, which X is going to be inactivated. It's not always going to be just the mothers or the dads. It's going to be a completely random occurrence. And so you can see that sometime in animals where the fur color, for example, is dictated by some genes on the X chromosome. So these type of cats that you see with like orange and brown mixed together, that's because of that X inactivation at random. So another thing that you have in there is that, these patterns, as we talked about, are going to be inherited in a very kind of stable manner. You have to first of all, inactivation happen at a very early stage during embryogenesis so that you don't mess up the regulation of developmental genes that may be present on that X chromosome. And this is controlled through two different things. One of them is there's a master transcription regulator that is present in our genome, and that is transcribed early in embryogenesis, and it is going to control whether or not certain genes are switched on or not. And so they can control, these transcription regulators can actually be controlled just by the process of embryogenesis happening. The zygote forming will automatically switch on certain transcription regulators. And then those are going to then regulate their own gene expression. So they are going to be made and kind of positively, in a positive feedback loop, keep making more of themselves until they're no longer required. And so this transient expression, so you can see that this particular gene A originally is going to be switched on because of certain developmental stage. Once it's switched on, it will act as a transcription factor for itself and kind of regulate its own propagation. So you will keep on getting more and more of this particular agent. And this gene A is going to then regulate the downstream activation of other genes that are going to control whether or not a particular trait is going to be switched on or off. If you don't have any other signal coming in, that's the way it's going to happen. Sometimes you're going to have another signal that will come switch on during development, and that second signal will then shut down this gene A production. Until that happens, gene A will continue to propagate itself and continue to produce it, and all the cells that are going to replicate and divide during that time will have the same exact genotype happening. So it's cell's own memory of what it used to be, right? So if you are a skin cell, you have certain master regulators already at play inside you, and they are going to keep propagating the same way in every subsequent generation, unless a differentiation signal comes in and stops that from happening, right? In which case, then it would switch lineage to a different setting. So a cell has essentially a memory of itself being a skin cell or a liver cell or any other type of cell that it is derived from. Another thing that it controls is what we call imprinting phenotypes. And so those are specifically methylation of these cysteine residues. Anytime you're methylating the DNA itself, not just the histones, but actual DNA methylation. So if you're imprinting or if you're methylating the proteins, the histones, you're putting the methylation on the arginine or lysine residues, right? So amino acids. When you are methylating your DNA, that is going to be on the cytosines. So it's going to be on these Cs, right, inside the genome. And those are not just any Cs, they're specific Cs across the genome. And those can be different in different individuals. But they actually control whether that particular gene is going to be expressed in the progeny, regardless of whether it's dominant or recessive. So it's something cool that happens in them. So these are specific cysteines. If you have unmethylated cysteines for a particular gene, that gene is going to be expressed in the daughter cell. And if you have the methylated cytosines in those particular gene, you are going to have silencing of that particular gene allele. Yes.
So it'd be like an ATC and then the C was just methylated and that would just not express.
That would, yeah, and there are specific ones across the genome that get methylated. If they are methylated in a particular gene, that whole gene shuts down.
Oh yeah, because
Yes, exactly. So why do I have them on here, by the way? He knows why I have them. Tell me. Tell us. Yes, exactly, because, yeah, our werewolf there imprinted on Bella's child. But so there is imprinting that happens in animal kingdom, maybe not quite, but it does happen. And we have plenty of examples of that, right? In birds as well as in animal kingdom, where you can you have that happen. And that is partly regulated by something like this, where you have methylation of specific genes that is faithfully transferred from parent to progeny, and that controls what signals they receive and respond to at the end of the day. So yes, so these kind of methylations are done by what we call maintenance methyltransferases that are going to faithfully take it from that original parental gene transcript, that particular one, and they're going to transfer it to the complementary sequence so that it is always the same ones that are transferred over. Okay. So, like I said, these particular modifications, both at the level of histones and gene, are going to be faithfully inherited by the daughter chromosomes and by the progeny that you get at the end of the day. So these parental nucleosomes with the modified histones, initially, you are going to have these transfer during the process of replication, so that half of the daughter nucleosomes are going to be inherited from the parent side, right? Parental, because that's the side that you have. And the other one is going to be the new side that's not going to be methylated. While these methyltransferases come through and they recognize which ones are done, and they come and do the complementary strand as well. So you can have that faithfully transferred all throughout the genome. So that is, again, another level of you regulating gene expression is at the level of our chromatin itself. And they are going to be modified two different ways: methylation, acetylation, or phosphorylation of the histone tails, or at the level of the gene itself. DNA itself can be methylated, acetylated, phosphorylated as well, but methylation specifically is responsible for imprinting as well as gene expression control. Now this can happen gene expression. Genes can be controlled by one single master regulator, or you can have multiple proteins that are required to control expression of a particular gene. So here you have an example where a single regulator can control expression of many genes. So an example from something you guys are doing in your lab would be, for example, P53, tumor suppressor gene. P53 can act as a transcription factor for a bunch of different genes that regulate either DNA repair pathways or apoptosis or DNA cell cycle regulation. And so as long as the gene has an operator region that has a P53 binding site, it's going to be regulated by the same transcription factor, right? And all of them have very varied function, right? One of them is causing a cell arrest, another one is causing your DNA to be repaired, another one is causing death of the cell, but all of them are controlled by the same master regulator, in that case, P53. So there are similarly many master regulators at play during development as well that control expression of either a small or a larger group of genes that are all responsible for then eventually making the phenotype that you're looking at. So here's an example where you have some genes that are expressed at a low level when there is no master regulator around. So you have this cortisol receptor that cannot bind to these genes in their operator region unless it is activated by presence of cortisol. So in the absence of cortisol, you will have some basal level of those genes being expressed, but it's going to be at a really low level or no level, right? Either they won't be expressed or expressed at a very low level, because the regulatory sequences for the cortisol receptor complex are not activated, since the cortisol receptor cannot activate them because there is no cortisol around. Only when you have the cortisol around, it's going to bind to the cortisol receptor. That in turn will activate it because there's going to be a change of conformation. It's going to allow this now new activated receptor to bind to these regulatory sequences for the cortisol receptor complex. That in turn will now activate these genes fully so that you have a higher level of transcription happening of these genes. Does that make sense? Yeah. Yes.
This might be a dumb question, but what is imprinting?
Imprinting is when you are inheriting either traits or gene expression control from your parent, and it's regulated not because of the type of allele that you have, but because of the methylation or these post-transcriptional epigenetic changes that are happening on top of them.
So there's traits.
Inherited traits, but not controlled by the actual allele, right? Like I said, so you could have, for example, let's say you are able to, you have, your mom and your dad. Your dad has the ability to solve some kind of puzzles really, really quickly, right? Quick puzzles versus this one does not, they don't have that ability. And let's say that this ability to solve those puzzles is the dominant one, right? So if you get a gene like that, you're going to be able to, even if you have one allele, you will be able to solve puzzles quickly. Well, if this is imprinted by methylation, then even if you get this gene, you're not going to be able to solve those puzzles because you had those, it was imprinted to be shut off. Okay. So it is kind of a second level of control where even a dominant allele may not be expressed because of these post-transcriptional, or not post-transcript, but because of these epigenetic changes. But it can also control, like I said, these really cool imprinting behaviors. For example, ducks being able to know how to travel a particular direction in winter. It's all due to imprinting genes and imprinting within their genome. Okay, so the advantage of this type of regulation, where a single transcriptional regulator, a master regulator, can control many, many different genes as a whole, as a group, allows us to have just a few master regulators and get lots and lots of different cell types. So at the end of the day, our bodies are composed of hundreds of different types of cells, right? And those are regulated just with a few, just a handful of master regulators. And the reason for that is that early on, again, if you look at the original zygote, during cell division, at one point, you're gonna have, let's say, one regulatory protein getting made, and that regulatory protein is going to start to diverge that particular cell into a different type of cell, right? And while this one continues to grow the same way, at a second level, when you have the second regulatory protein you're getting built in those, whether or not the first regulation was there will dictate what type of cell type it makes. So just two regulators here are able to convert these into many, 4+ cell types, not just two different ones. So it's the combination effect from different master regulators that allow you to get lots and lots of different cell types within a very short regulatory period. Okay. So a master transcription control gene can control not just gene level transcription, but also organ level transcription. So if you look at, again, embryogenesis and development of different organ systems and different types, right? You have those 3 layers that form initially that are going to give rise to all your internal organs and all your different parts of the body. Well, at that level, it's just 3 different genotypes, are essentially 3 different gene expression controls that are happening. But because of that, each one of these is going to be really, really important in a particular type of organ system or a particular type of development. And so Drosophila, in Drosophila, if we have this particular master transcription regulator, EY, triggered in any cell type, during embryogenesis, it will develop an eye artificially at that point. So that's how powerful that control is, that just this one gene, this one master transcription regulator is able to control the production of an entire organ, right? And so similar systems are present in humans and other more complicated organisms as well. Another function of these transcriptional regulators is to allow cells to get differentiated in general. And for those, these transcription regulation can not only start from an undifferentiated cell and convert it into a differentiated cell, they can also sometimes take a differentiated cell and convert it into a different type of differentiated cell. This is extremely important nowadays when we are looking at research to try to produce organs of our own to transplant, right? Whether at a simple level or a more complicated level. But that is essential for us to have enough organs to be able to help those that need them. But these transcription regulator, for example, you could have cells from the skin, right? But if given the differentiated environment, changing the gene expression inside it, you could take those same skin cells and you can turn them into neurons or muscles or anything else that you want, just by knowing what transcription regulator or master regulator would be responsible for those actions. So in our cell types, for especially, for the, in general for mammalian animal kingdom, but humans specifically, there are these 3 master regulators, KLF, SOX2, and OCT4, that combined give rise to pretty much all the different cell types that we have in our body. So these transcription regulators, we can control at different levels to get the type of cell that we want to get during the process of differentiation, examining those effects and getting what we want from there. So here is an example of how you would use it in research. We start off by using what we call pluripotent iPS cells. So does anyone know what the word pluripotent means? So this is looking at a type of stem cell technology. So how are pluripotent cells different than a typical, let's say, a zygote or a stem cell that you get from early development? How are these different?
Can change.
A zygote can change into you.
I'm talking about these types of cells can change into different types of cells.
Yes, they can, but what can they not change into?
Back to their original one.
Let me go back to who they are to begin with. So we talk a lot about like ethics in stem cell technology, right? You hear that all the time, whether stem cells should be allowed or not allowed. What's the biggest problem that people have when it comes to ethics of stem cell technology? What is it that they don't want? Huh?
This is, there's something like that.
So partly it is, taking something from these fetuses, but if the fetus wasn't viable, that's not really that big a problem. What is the bigger problem there? What do they think they can get from those cells?
Oh, maybe they cannot like make a human?
Yeah, so their idea is that by taking these cells, you are now stopping that individual from becoming a human, right? But the stem cells that we use primarily in research are pluripotent, not totipotent. A totipotent stem cell would be a cell that can give you the entire organism as a result of differentiation. A pluripotent stem cell can give you multiple cell types as a byproduct of differentiation. It can never give you the entire organism. And that's a very, very important thing to take from this. That the stem cells that we are looking at here, in most cases, when we are doing stem cell research, is using cells that are not totipotent, that are pluripotent. And so they can be made into a subset of cell types within the system, but never be made to get the entire organism. And we'll talk about when and where you can get those from. So there are these, like I said, 3 master regulators that we can use to create structures like that. So fibroblasts are these cells that are present in your extracellular matrix, right? And they're very easily propagated. They are very quick to propagate and multiply and replicate. And they're kind of very basic differentiated cells. They're not very differentiated, but they're differentiated cells. And these cells can be induced. This is one way that you can get a pluripotent cells. They can be induced to de-differentiate, right? Go back to its de-differentiated form or undifferentiated form by introduction of these genes into their nuclei, either artificially or through activation. And when you do that and allow them to divide in culture, they kind of return back to a more pluripotent system where they can be made into multiple cell types. And these are what we call these IPS. These are induced pluripotent cells. Yes.
Are the stem cells, so I know that you have stem cells in your gut lining to replace your gut lining, are they pluripotent or are they?
Yes.
Okay.
Each organ system in our body, including nervous system, by the way, has a small population, in some cases small, in some cases a little bit larger, population of pluripotent stem cells that can be used to make cells related to that particular organ system. So typically, if you are looking at, for example, a neuron, right, and you're looking at a brain stem cells, they can only be used to create one of the neural cells, right? You can make glias out of them or astrocytes out of them. You cannot make just any cell, muscles out of them. But then you have populations elsewhere, for example, in bone marrow, that may be a little bit more Yes, pluripotent. Yeah.
So if we take OCT4, SOX2, and KLF4, we put that into the fibroblasts, and then we turn them into IPSs. And then we put OCT4, SOX2, and KLF4 into them to differentiate them again into whatever we want them to be.
So you can't just use them alone to differentiate into different cell types. What you do is then provide them with the right environment associated with a particular structure. And then they will induce, that second level of induction is partly from environmental control. So there are then other genes that are going to be expressed that are going to be just present in a muscle or in a nerve or in a, hepatocyte, whatever the case may be. So here you have these IPS, these induced pluripotent stem cells, and you can control them and induce them to differentiate in culture into smooth muscle cells, into a neuron, into a fat cell, into a skin cell, whatever you need, based on your knowledge of what is needed to differentiate into those cells. So we know clear differentiation factors that are required for many of our muscles, of many of our cell systems, and we can use those conditions in culture and those agents, those differentiation, provide them with those differentiation signals artificially, and then they will induce them into those systems. Yes.
So earlier you said, if this becomes a pluripotent cell, it could go back to its original state?
So this one, we could differentiate it back into a fibroblast if we wanted. It can never be a human. That's the most important part to take from it. It can only be differentiated into a certain types of cell, different categories. It is able to turn into multiple different types of cells, but it can never give you an entire human by itself.
Right, but once it's turned, can it go back?
Yes.
Okay.
Definitely, you can go both ways.
That's something so they can partner related, like or heart or whatever. Very potent stem cells
Yes.
from those environments.
Okay, so that's, and we'll come back and talk about more with the differentiation control for this too. And this becomes important for those of you who are examining differentiation of maybe meioblast or neuroblast in the lab for your poster project, looking at those differentiation, that is part of the process. What you will be providing to them in the form of those supplements would be potential differentiation factors to see if they are able to do those effects or not. Okay, so next we're going to talk about some post-transcriptional controls that are needed in order for us to regulate our gene expression. Again, so you are regulating gene expression at every single level, right? Before transcription ever starts, just at the genome level with the chromosome modifications, you again regulate them at the promoter region, right? At the transcription level, you regulate them at translation level. You can also regulate them at post-transcription and post-translation level. At the post-transcriptional level, you are going to be looking at the RNA that you can produce from that particular gene. So obviously we've talked about RNA splicing already, which is one way that you can control what protein isoform is getting built from a particular transcript. If you remember for the beta globin gene, we talked about how it can be differentially spliced to give you different types of protein isoforms and different cell types. You can also look to what we call small regulatory RNAs. These are small sequences that can lead to silencing of their complementary gene expression levels. So these are microRNAs. They are small sequences that are going to be complementary to your mRNA sequence. And what they do is they can bind to your mRNA itself. And when they bind to your mRNA, they create small regions of double-stranded mRNA essentially, right? And that double-strandedness of the mRNA will signal to the cell that there's a lot of this present and it can be shut down. And so it will destroy any transcript that's left behind. And another way that we can do that is by using foreign RNA sequences against so in molecular biology, that's what we do. If I have a gene that I'm looking at its function and I want to silence it, right? And I want to make sure that my effect that I was observing is in fact from that gene expression, I can create small mRNA, microRNA of my own using a sequence against the mRNA at some point, and I can transfect it into the cell that I'm looking at. And this is called RNAi or RNA interference, and it will shut down the expression of that gene either partially or completely. And then I can use that to confirm my observations about the function of that gene. So in bacteria, you can have another way of this happening. And this is where in bacterial gene, you can have these, again, stem loop structures that can form in their regulatory region. We already saw that in tryptophan, right? That was exactly what happened. That in tryptophan, similar to that, in the mRNA, in that untranslated region, you can have these stem loop structures. If you have those, they are going to either block the start site directly or indirectly. And when they can directly block it by just existing like this, so the polymerase can bind. Or sometimes you have translation repressor proteins that come in and bind to the start site instead of them. And they can bind to these regulatory sequences. And if they are bound, you can't get actual translation of that transcript, so no protein is made. Yeah. So here is, again, the same thing. You have up here, one of them is by actual translation repressor protein binding to these stem loop structures and blocking the start site. Another is just the stem loop structure itself is going to be made in a way that it cannot allow for the polymerase to bind. Now, sometimes this happens because you have too low of a temperature, for example. So heat shock proteins or some other proteins can have these structures that prevent the start of protein translation. But if you increase the temperature, these stem loop structures typically come apart, and then you can have those sites open and you can have proteins getting built from those particular sequences. Yeah. So microRNA, basically, what do? They target their complementary sequences on mRNA. When they bind to those sequences, they create these double-stranded area on the mRNA. This causes other proteins to recognize, this is how the cell knows that they need to shut it down. And these proteins are called RISC proteins. These RISC proteins recognize these areas of extensive match, and they are going to lead to rapid degradation with nucleases that are within the RISC proteins. And now the transcript is destroyed and it can't obviously be translated or done away with. And this is a quick way to control transcription of a particular protein. Let's say you already have this protein around and you need to shut down production of any more of this protein. This would be a very quick way to do it, right? You can just create these microRNAs that can bind to any mRNA that's present. It's going to cause it to degrade, and then that's going to leave no mRNA transcript around for more protein to be made. Yeah, question? No. Okay. When we add foreign double-stranded RNA, like I said, RNA interference or silencing RNA, we can call these structures. We are providing artificially small double-stranded sequences of that mRNA. And again, they are going to be recognized by RISC proteins, and they're going to be like, oh, there's a lot of this around, kill all of it. And it's going to destroy any transcript related to that silencing sequence. Yes.
So the RNAi has the same exact, or not the same exact function, but very similar functions to the microRNA.
It's the same thing, but artificially produced. Yeah. So the silencing, these are foreign double-stranded RNA. We made them and put them in, as opposed to the cell itself making them. When the cell makes them, it's the microRNA. When we make them and put them into the cell, it's silencing RNA. Cool. So yeah, so they can do, like I said, they can obviously destroy the transcript, or they can also directly silence the gene expression, because they are going to bind to these transcripts that are coming out of your actual genome. And when they bind to it, that's going to negatively impact that particular transcription from happening further. So they can do it both ways. Another thing that we can have are what we call long non-coding RNAs. An example of that would be that leader peptide that we saw in tryptophan. So these are sequences that are coming out of either the untranslated region or otherwise in the genome that produce an mRNA or an RNA transcript, but that is not part of translation sequence. It's never going to be translated into an actual protein. And these long non-coding RNAs can bind directly to other proteins. Can you give me an example of that, where it can serve as actual function? There are lots of them. So non-coding RNA. Where would you see them? So like I said, I give you one example that they could create these leader peptides, right? That can have regulatory function through that. What else? You know, of many long non-coding RNAs that have very good functions, very important functions in our genome. Yes.
That's a box.
Not the Tata box. Poly-A, not the poly-A tail. Come on, yes.
Sequence.
Well, those everything that you're talking about is DNA, all these Tata box, but you're getting closer to it, yes.
About the RNA that binds to the telomere?
Exactly, that's one example. Where's your sheet, man?
Oh, okay.
What else? I want somebody from here to tell me. We talked about so many sequences that can be used that are long non-coding RNAs that have many functions inside our cells. Yes?
The RNA primer, like when you're coding for DNA with RNA.
That's a tiny little, that's not a long non-coding RNA. But, so ribosomes, what are they made of? People back here. What are ribosomes made of? Protein and? Yes, they are made of RNA. All those RNA molecules that are embedded within the ribosome that are essential for it to recognize the sequences and appropriately take other agents to make it, what are they? They are long non-coding RNA. They're not coding for protein, but they are coding for, that's it, they are doing work. What about tRNA? Can you call that a long non-coding RNA? Yes, right? tRNA itself is a long non-coding RNA. It's not coding for a protein, but it is a functional RNA molecule. And it is essential for protein translation. Yes. No, mRNA is not a non-coding, it's a coding RNA. mRNA is an example of not a long non-coding, but the opposite. Yes, ribosomal RNA, this is what she had said, exactly. Any questions on this? Yes, no, maybe? Okay, we're actually gonna go ahead and start on our next one a little bit. Before class ends today. Because it kind of flows, you know, sometimes our next lecture is a little bit off and we don't wanna mix them up, but this one actually flows very nicely with what we were just doing, so. By the way, she explained cell signaling so beautifully that I would just love for you to listen to her. You might like her even better than me. I don't know if you like me. Even if you don't like me, you would love her. How about that? And then you'll be like, why don't you teach more like her? Because I don't teach it. But that's okay. She's amazing. I love listening. I'm sorry. I'm easily amused, I guess. Okay, so next lecture. So these, you know, right now we are at the core of molecular biology. So the set of lectures that you have for this particular exam, we are probably the hardest of the material that you're gonna get. And then when we get into the last set of lectures, you'll be like, oh, this is a piece of cake. compared to this one. So just be aware, this one is the hardest of content. So what are we gonna talk about next is cell signaling. So I'm gonna touch a little bit today on general principles of cell signaling, and then we will continue it next class and go talk about various types of cell signaling more in detail. Two specific ones that we'll focus on are G protein coupled and enzyme coupled receptors. So what exactly is cell signaling? Cell signaling is essentially the way the cell responds to changes within itself and around itself. So it's a way for it to receive signals from outside and then use them to change its behavior, its function, its properties, to match it, right, in order to survive in that environment, and also how it acts on things that it has already happening within it. So it is a way for it to respond. Now, this cannot happen transferring that signal in one way, shape, or form to the nucleus where you have the main command center of sorts. And this can happen sometimes as an extracellular signaling molecule binds to a receptor on top of the cell, but it can also by the molecule going into the cell and then binding to appropriate receptors inside the cell. And we'll have both of those things that we will talk about. So here are a few things that signals will allow the cells to do. In a typical environment, the basic living being has to survive, right? So it receives signals, it has to change its behavior so that it can continue to survive. That's the most basic level of existence. The next level up, it needs to be able to grow and divide, not just survive. And so in this case, it needs to be able to assess the environment. It needs to be able to make sure that it has the space and the environment in order to grow and divide, and then be able to respond to it internally and control it in some way so that when it is able and when the environment is amenable, it can grow and divide as needed. At the third level, you have cells that are now not only surviving, growing, and dividing, but actually doing important works for that entire organism to exist in the environment. And in order to do that, your cell needs to be able to specialize. And to specialize, again, it's going to have some internal signals that will help it differentiate into appropriate cell type. It can also respond to external signals that will tell it what type of environment it needs to create and in what way it needs to differentiate. And finally, a living cell needs to be able to recognize when it is time to go. And that could be in the form of external or internal signals that recognize when it is either too old, too damaged, or unneeded. And in any of those conditions, you will have that cell eventually die out. If it dies through a very programmed cell death process, it will be through apoptosis, but that is not the only way they die. They die other ways as well. But we are going in a cell-dependent, extracellular signaling dependent mechanism. We will talk about apoptosis specifically as the way a cell dies, because that is the programmed cell death mechanism. Now, the same signaling molecule can have very, very different effects, and it is going to be dependent on what type of receptors binding partner it finds or what type of receptors it binds to. So the same protein molecule, acetylcholine, or the same small molecule, acetylcholine, can bind to receptors both on heart pacemaker cells as well as in salivary glands and in skeletal tissues. But depending on which type of receptor they bind to will lead to a very different response. So in the case of heart pacemaker and salivary glands, the actual receptor protein is the same, but the internal environment is the different. So in those cases, you have a different response. In the case of heart pacemaker cell, it's going to lead to the firing of signal to create the heart rhythm inside those pacemaker cells, right? With the heart beating. In the case of salivary gland cells, that cell cannot contract and relax and create a electrical output. Instead, what it does is that it releases certain secretory molecules in response to the same signal coming in. So it's the same idea that it's kind of contracting and relaxing in its own way, but it is doing a very different response. This is going to lead to the secretion in this case. And then in skeletal muscle cells, the receptor is different, and that receptor allows the cell to contract when acetylcholine is present on those, bound to those receptors, okay? So again, same molecule, acetylcholine, but depending on the binding partner, leading to very, very different responses. They can also act, so the other thing is that signaling molecules don't have to be made by the cell itself or don't have to be right there nearby. You can actually have molecules far, far away in a completely different organ system. As long as they can make it to the target cell in one way, shape, or form, they can have a response. Right, and in our body systems, in our multicellular system, it's the bloodstream typically, or the lymphatic system that is going to carry these molecules over. But you can also have them happening through the neuronal channel, right? So you can think of the endocrine cell producing a hormone, going into the bloodstream, going to the target cells where it binds to receptors and have an effect. You can think of paracrine signaling, where the cells are right there in their environment and they are producing signals that the cell is responding to. An example would be a cell, let's say a cancer cell in a organ system, and it's beginning to grow and divide, and now it doesn't have enough blood supply. It's going to produce small molecules like VEGF, which is vascular endothelial growth factor, and that signal is just going to act on blood vessels nearby. Right? Because it doesn't have any other way to talk to them. So this molecule is going to get secreted from the cancer cell. It's going to act on nearby blood vessels and induce them to grow towards it. And that process is called neoangiogenesis, but that's an example of paracrine signaling, right? But other cases where hypothalamus, for example, or pituitary gland are producing certain hormones and they are affecting your growth or they're affecting your temperature regulation, those would be example of the endocrine system controlling far away other body systems to do the work they need to do. Another one would be the neuronal signaling, where you have a impulse fired through our nerves in order to affect a target cell. And that would be in the form of neurotransmitters that are released by that system at the end to the target cell to have the response they need. And finally, in some cases, you have to be bound to the signaling cell in order for it to respond, for you to respond to it. In that case, the target cell may be, can you think of an example of that actually before I give you one? Of a contact-dependent signaling. That could be an example in a way where a viral particle binds to a receptor and causes an induction of a response. Typically, this would be in the extracellular matrix. There would be certain cells and proteins that are binding to your cell, and they have to be bound to it in that location in order for the cell to have, for example, cells have this, abnormal cells will have this ability called contact inhibition. When they are bound on all sides by other cells, they will not grow and divide. That signaling will only happen if there are cells bound right there around it to itself, right? So that would be an example. Okay, so the second part is that how do we respond? How do we actually receive these signals? We talk a lot about cell surface receptors and them acting as your signaling molecules, right? So the signal is binding to the receptor and then leading to a change inside. We'll talk a lot about those, but that's not the only way. Many agents actually pass through the membrane into the cell and bind to a receptor inside. It could be a receptor in the cytoplasm. For example, retinoic acid is a small molecule that crosses through the plasma membrane into the cytoplasm. It binds to retinoic acid receptor inside and then gets translocated into the nucleus, acts as transcription factor for differentiation. So that would be an example for those. So you don't have to bind to the outside. You can actually internalize the signal and then bind it to a receptor and have an effect as well. So we're gonna stop here and next time we will talk about all kinds of receptors and what they do and how they help us differentiate, grow, and develop.
I know.
Only got 3 paragraphs on.